Thank you for coming to my talk. So I have been a professor at UCLA for 12 years, and I just started a new position at Fred Hutch. So I'm very fortunate to be able to still speak here, and I have been attending CGSI for a couple of years. So this time I'm going to share a new work by my postdoc Pan, who is also in the audience, and the work is called mcRigor. So what it stands for is that MC stands for Metacell, and we hope to make this Metacell partitioning, which is a common practice in single-cell data analysis, to become more rigorous using statistics. So first of all, for our students, I want to first briefly review what single-cell data analysis is like. This is a figure from this paper, which shows how the data is analyzed. So as we have heard from the previous talk, for single-cell data, we can measure omics in every individual cell. So you start with sequencing and you do some cell barcoding. So that allows you to allocate your reads to different cells. And after some pre-processing, you will get this matrix, a gene by cell expression matrix. But what you don't have is the cell's identities. So you need to do cell type annotation by some clustering and to find highly expressed genes in each cluster and using those markers to annotate cell types. Besides, you want to identify some regulators, which we heard from the previous talk. So besides those analyses that at the cell level, you may also want to do some analysis at a gene or feature level. So this is a figure I found from a paper which tried to do some gene-gene co-expression or correlation analysis. But one thing you can note here is that a lot of the counts of a gene are zeros in many cells. So this is known as an issue called sparsity. And you can see that in this paper, the authors use some way to actually transform the data, so then the two genes look very positively correlated. However, how trustworthy is this step? I think that is a question. And this is a paper that tried to do some gene regulatory network inference using single-cell data. So with those data transformation, they do some downstream network construction and also network-based analysis. But the first issue here is sparsity. So this is a well-known issue since the inception of single-cell RNA-seq in 2018. And my group previously developed one of the first imputation methods for this problem called SE-impute. But later, we and other people found that imputation is not for free. There's no free lunch. You need assumptions so you can actually recover the zeros you think are false and change them to be non-zeros. But what assumptions are reasonable, that matters. So therefore, people have criticized imputation for introducing artificial signals into the data. So in parallel to imputation, more recently, a practice has been done in many single-cell data analysis called Metacell. So from our perspective, in terms of statistics, this is a heuristic solution. So let me just describe how it works. Essentially, you define metacells by clustering single cells. So each metacell represents a small cluster of cells. But here we should not confuse the clusters here with the clusters we did for identifying cell types. Because for cell type annotation, the clusters are big. You want to use each cluster to represent one cell type. But here, every cluster as one metacell is very small, typically from 10 to 50 cells. And what you're doing with them is, with each small cluster, you take the average gene expression profile, and afterwards, you will work with those metacells instead of single cells. As you can imagine, such aggregation would largely reduce your zero proportion, because as long as one gene has a non-zero value in one of the cells included in the metacell, then this gene will be non-zero in the metacell profile. So another advantage here is that with this aggregation, your metacell number would be much smaller than your single cell number. So you can also reduce the computational burden. But as I said, it's a heuristic solution because there's no way to know for sure how large each metacell should be like. So the question here is, what is a reasonable definition for aggregating cells into metacells? You can think about the two extremes. One extreme, you aggregate all cells into one metacell, and that is called pseudobulk. Another extreme, you don't do aggregation at all, and every single cell stays as a single cell. So if you believe that pseudobulk loses single cell resolution, not so ideal, and single cell data is too sparse, then there must be some sweet spot in the middle. So before our work, there were 4 Metacell methods developed in the field. First method, Metacell, and then by the same authors, Metacell 2, try to use parallel computing to speed up. And then SuperCell, a separate method, and then SEACells. So this one was from Dana Pierce's lab, published in Nature Biotechnology, and received a lot of attention. So more recently, there's another method recently published in Nature Communications called Metacell. So besides those standalone methods that can work with any data, we also have some high-profile papers that did data analysis, and they implemented Metacells in their own way. So they use the practice. However, if you look at how they constructed Metacells, there's no consensus. So let me just show you one example from a review paper that shows that if the Metacells are not defined well, your analysis could be biased. So the top row shows the single cell data, the bottom row shows your metacell data. Suppose that you know the single cells belong to 2 types. If a metacell you construct happens to contain single cells of different types, like this one, then including this metacell in your gene-gene co-expression analysis, you may have a false positive correlation between the 2 genes. However, if you look at the single cell data, maybe the 2 genes are not so co-expressed. They are probably exclusive. When one gene is expressed, the other gene is silent. But with the dubious metacell, then you can have a false conclusion in your analysis. So that's why we think it's very important to identify those metacells if you want to use metacells. So in our work, we want to answer the following 3 questions. First, how to define a metacell. Second, how to detect dubious metacells. Third, how to optimize metacell partitioning, given that we have 5 methods to choose, and also each method has its own hyperparameter. How do we optimize? So Pan's work called mcRigor is already available on bioRxiv for a while, and it has been published accepted by the RECOM conference this year, and also accepted in press at Nature Communications. So to first question, how to define a metacell, we go back to the first publication called Metacell to look at how the original authors proposed the definition. This is a quote: "A metacell is a homogeneous collection of single-cell profiles that could have been resampled from the same original cell." So how do we interpret the sentence? We translate it a little bit into statistics. So we think it refers to the fact that if you look at a gene's variation across the single cells within a metacell, these differences, the variation, should be attributed to measurement error instead of biological signals. So this translation reminded us of this paper by Matthew Stephens, published in 2021 as a perspective paper that tried to decompose how we model the statistical model for single-cell RNA-seq data into 2 layers. One layer called expression model, which is about genes' true expression levels distribution. So here it's latent, but we consider this to be the biological variation we are interested in. And the second layer on top of the first layer is called measurement model, which is a conditional distribution, the distribution of our data, observed counts, given the true expression levels. Mathematically, we wrote this 2-layer model. So we denote cell by ith cell, and we consider gene to be J. So J goes from 1 to P. So P genes in total, N cells in total. So lambda I is what we call the true expression level of the P genes in cell I. And we consider lambda I to be the expression proportion vector instead of the absolute abundance. The reason is we know sequencing experiments can never provide the absolute abundance, but only the relative abundances of the genes. So therefore, lambda I is the true proportions of the P genes, which is assumed to follow some P-dimensional distribution. We don't know. On top of lambda I, given the proportions, so lambda ij is basically gene J's true proportion in cell I. How do we get a count for our sequencing reads, or the unique molecular identifier, UMI, Yij? So this is a count. So this is actually typically modeled by a multinomial distribution. So the total count yi plus is called library size of cell I. We allocate this total count by the proportions to get the counts, subject to some stochasticity in the assignment. So if we believe this two-layer model, then essentially, if we think a metacell represents just one biological state, and the cells in this metacell only differ by the measurement model, then they should share the same lambda, and their differences is only attributed to this allocation step. So then we come up with a statistical test. That is, we can check each metacell to see whether they satisfy this definition. If that's the case, we call this metacell trustworthy. Otherwise, we call the metacell dubious. So here's our proposal, mcRigor. Our goal is to provide a statistical criterion to identify dubious metacells that are already constructed, consist of single cells from different states. Besides, we want to nominate the top performing metacell method and optimize its hyperparameter. The key hyperparameter needed for every metacell method is called granularity level, gamma, which is the average size of metacell. So in other words, you divide the number of single cells by the number of metacells. That's the average. But please note that every method has its own way of defining metacells by clustering. So the metacells are not of the same size. They can have different sizes, but this gamma is the average. So how can we achieve the two goals? Our intuition here is based on the two-layer model I just described. Within a metacell that is trustworthy, the features should be approximately correlated. So this is just an illustration of the gene-gene or feature-feature correlation matrix. We believe that if the cells' differences are only due to the measurement model, the correlation matrix should look like this. So close to be a diagonal matrix with some spurious off-diagonal entries but no pattern. The reason is we do not think the technical measurement noise should drive gene-gene correlations. On the other hand, the right matrix, gene-gene correlation matrix, may indicate that the metacell is dubious because here we see some block correlation structure that may indicate that the genes are correlated because of their biological signals, not just due to the technical difference. So in other words, we can compute one such matrix for each metacell and check whether the matrix is close to a diagonal matrix. That's the intuition. So for this, we propose a per metacell statistic called MCDIV, stands for metacell divergence. And how can we set a threshold on the statistic? This is the key part. And Pan came up with this very interesting idea to use permutation to generate null values for the statistic, but we want to preserve cell library size. The reason is that going back to the two-layer model here, you can see the cell library size is a key factor here in this multinomial distribution. Basically, the genes would have some small correlations due to the multinomial, but this is given the cell library size, so we want to preserve this. Okay, so our first idea is trying to permute each gene on its own independently so that the genes become uncorrelated. So we can create a scenario where the genes are uncorrelated, like the null case. And this is an idea we implemented in the previous work called SED for visualization. I'll quickly give a snapshot in the last slide. But we started with this idea. So you may want to ask, why do we need to do permutation instead of doing some well-established statistical test for this statistic we propose? The reason is that here within each metacell, the number of single cells is not so big. So like I said, 10 to 50. So the classical statistical test that relies on asymptotics, like n goes to infinity, would not be applicable. So that's why we need to come up with some ideas that can work with small sample size, and permutation is one of them. So we started with a within gene permutation, like I just showed. You did remove gene-gene correlations, but you also removed cell library sizes. So after the permutation, these permuted cells do not have one-to-one correspondence with the original cells. So you lose the cell identities here. So that's why this is not so ideal. How about the other way? We do within cell permutation. That is, we look at all genes values and shuffle them within each cell. This way can allow us to preserve cell library sizes. And after this permutation, these new columns will also be uncorrelated, but they are no longer corresponding to the original genes. We call them permuted genes. So in other words, this within cell permutation can allow us to preserve cell library sizes, but we lose the gene identities. Then what do we do? So here, Pan came up with the idea. So we can actually provide a normalization factor for each set of genes. So in other words, in the normalization, we do within gene permutation for each. So here we actually permute each gene in the original gene set. And down here, we permute each permuted gene. So this is called double permutation. And so after each permutation, we can create a normalization factor to normalize the fact that here we have different sets of genes. That's the intuition. So using the top row, we came up with a statistic. Using the bottom row, we came up with a null value for the statistic. So now let me give you more details about our method. For one metacell, we look at the original data, calculate gene-gene or feature-feature correlation matrix, take its difference with the identity matrix. So we are interested in the off-diagonal entries, right? And we take the Frobenius norm to summarize the off-diagonal entries. This is the numerator. In the denominator, it is what we call the normalization factor. We take what we get from the within feature or within gene permutation to calculate the resulting gene-gene correlation matrix, do the same difference. And then we take the ratio. That's our statistic. In the bottom, we calculate the null value for the same statistic, but we did within cell permutation first. So we can preserve cell library sizes, but remove gene-gene correlations. But here, since we're working with permuted genes, we need its own normalization factor. So we get a null value for each metacell. So we do this for each metacell, and we pull them together. So the null values will give me a distribution, using which I want to set a threshold for my actual statistics for this metacells, so I can do some thresholding. So in detail, here is I'm getting. So pulling the null values, I noticed that the null values have some relationship with the metacell size. So we plotted this trend, metacell null values versus metacell size. So the size is the number of single cells within. So we can see that by sliding window, we can get a conditional threshold on the null values given the size. So we get this 95 quantile function. And then applying this function to our actual values of the statistic, we can see some of the metacells values are above the threshold, meaning that they are too big, then they are bigger than what we expect, and we flag them as dubious. And for those under this threshold, we call them trustworthy. So this is our dubious metacell detection step. So let's look at some results, how they work. We first applied this idea to this analysis from the supercell method paper. So this method is a metacell method, and it was applied to this data set for COVID-19. Essentially, we have single-cell blood data for healthy patients and COVID-19 patients. We want to look at some gene-gene correlations. In particular, we're interested in the adaptive immune response genes. So the genes in this yellow box here. So if you look at the single cell data and compute gene-gene correlations, you don't see a much big difference between healthy patients and COVID-19 patients, even though we expect these genes to be active after COVID-19 infection. So in the supercell paper, they constructed metacells and using metacells to do the correlation analysis. So we can see that for adaptive immune response genes, their correlations become much higher, but still higher in both groups, and the difference is not obvious. So what we did is we used mcRigor to filter out dubious metacells for each set, and then using only trustworthy metacells to do the association analysis. Now we can see that this gene group is not so correlated in healthy patients, but correlated in COVID-19 patients. And then we look at how the Metacell data actually look like for those healthy patients. So for the 3 gene pairs here, you see the blue Metacell is the driver for the high correlation. But these are dubious Metacells. So that's why removing them, the correlations will get back to low. And looking at the original single cell data, we don't see strong correlations there. So supporting that removing dubious Metacells is reasonable. A second application is about the single-cell multi-ohm data, where single-cell RNA-seq and single-cell attack-seq are measured in the same cells. So attack-seq measures enhancer signals, RNA-seq measures gene expression signals. So for using the single-cell multi-ohm data, the question is, can we find associations between enhancers and their genes, nearby genes, so we can guess which enhancer might regulate which gene? So using single cells again, you see that we don't see many enhancer gene associations, only 4 edges here. And using all metacells, this is constructed by the SEACells paper. It's another metacell method, and we take their analysis and try to repeat it. Using all metacells, yes, we get more associations in the 3rd row. And using our mcRigor to again filter out dubious metacells, you see this blue, this red edge is gone, no longer here, but we added this blue edge. So we look at the differences. So for the 2 differences, we use the original data and the literature to confirm that the removal of the red edge seems reasonable because we actually don't see much ataxic signal in single cell data. But adding this blue edge seems reasonable because we do have some small signals here, and this is also a validated enhancer. And we also check the single cell data to see whether if we ignore the zeros, do the non-zeros tend to support our conclusion? And that's the case. So this is the second application. So besides detecting dubious metacells, another use of mcRigor is to optimize the granularity level gamma, the hyperparameter. So you can imagine that there is a trade-off. The smaller the gamma, it means that we have more but smaller metacells. So here, they are less likely to be dubious because each metacell is small, but you still have a high sparsity. That's the challenge you want to counter. On the other hand, if gamma is too big, every metacell is big, then you will have more dubious metacells, but of course, the sparsity level will be low. So there is a trade-off between the remaining zeros, we call zero rate, and the dubious rate, the amount of dubious metacells. So Pan came up with a score by combining the dubious rate and the zero rate. So we take a weighted sum, and the default is that they have the same weight because both rate ranges from 0 to 1. So 1 minus the total weight is what we call a score. The higher the score, the better. So in other words, you see that as we change the hyperparameter, this blue curve shows the dubious rate goes up. The green curve shows the zero rate goes down. And we have this combination here, which shows that in the middle, we balances the 2 rates. So on our simulation, we first found that we can find the right gamma in the simulation. On the real data, I'm just showing one example here. We have more in our paper, which is I found to be a very interesting example because this is a data set we used when we try to validate our imputation method. So this is an interesting data set where single-cell RNA-seq and single-molecule FISH, another technology based on fluorescence, are measured on the same cells. So in other words, single molecule FISH is not high throughput. You cannot measure the whole transcriptome, but you can suppose it to be more accurate than single cell RNA-seq data. So we use the zero proportion in single molecule FISH data as our ground truth. And we can see that as we increase the gamma for each of the 3 metacell methods, the zero proportion will go down as gamma goes up. But where should we stop, right? Using our mcRigor to pick the optimal gamma for each method, we look at the triangles. They are the optimal gamma for each method. And at these optimal gamma, the remaining zeros in the metacell profiles tend to agree well with the zero proportion in single molecule FISH data. This is an indirect validation to say that the gamma we chose is reasonable. So I will summarize by saying how we use mcRigor in practice. So there are 3 steps. First step, you can apply your methods of choice and the candidate hyperparameters in combination to a single cell data set. So each combination method and hyperparameter will give you one set of metacells. And you can apply mcRigor to calculate a score for each metacell partition. And you pick the one with the highest score. So this is what's considered optimal by mcRigor. And then, given this metacell partition, you run mcRigor again to detect the remaining dubious metacells and flag them. So then, the next step is you can throw away these dubious metacells and don't use them, or you can use another add-on we provided called mcRigor two-step. That is, you can decompose these dubious metacells into single cells and repartition them into metacells. So in our paper, this is what we added during the revision. We found that the mcRigor two-step can better preserve the rare cell types like this one and this one. So before, if we just throw away dubious metacells, then these two small cell types, rare cell types, will not have metacells remaining. But using mcRigor to do a second time reconstruction of metacells, now we can have some metacells for the two rare cell types. So I want to stop here by acknowledging Han for doing this work. And yeah, so we'll look forward to your comments, and hopefully it can be a useful tool to be added to your single cell data analysis. And at last, I just want to say that a similar permutation idea, but simpler, the within-gene permutation, was previously implemented in our other method for detecting dubious two-dimensional embeddings and optimizing hyperparameter for the 2D visualization using popular methods like t-SNE and UMAP. So thank you for your attention.